Toby Bell, comes to us from the School of Chemistry to tell us about imaging the nano world molecule by molecule. Toby began his studies locally at the University of Melbourne with a science engineering double degree, followed by a PhD, graduating in 2002. He then spent several years in Germany and Belgium, working as a postdoctoral fellow. In 2006, Toby returned to Australia to take up a centenary research fellowship and join Monash University as a lecturer in 2009. Toby is interested in discovering the fundamental photophysical properties of new materials at the single molecule level and using single molecule spectroscopy to look at biophysical problems like protein folding, aggregation and conformational change. Outside of the world of science, he also plays the clarinet in the Zellman Symphony Orchestra and also the Quinity Wind Quintet.
Thank you everyone for coming along. It's a great pleasure to give you a little snippet into the science that I'm doing in my lab. So I want to tell you today about a way that people have developed to improve and get around a problem in imaging using light. So the diffraction limit limits the resolution that you can build an image by using light. And if you build up an image molecule by molecule, you can actually get around that diffraction limit, and people so far have made images with perhaps an order of magnitude improved resolution. So fluorescence is a well-known phenomenon. I'm sure you've all seen it along the way. In case you haven't, got a little jar here with some fluorescent dye in it, and fluorescence is the phenomenon where the molecules in here can absorb light and then they will emit light. So if I just shine some light on here, so this is a purple laser pointer, we see some nice yellow fluorescence coming out from there. Just put it in like that, you can see it. And this. Okay, so fluorescence has been used for a couple of decades very successfully to do microscopy, to build up images. The key advantage is that chemists and biochemists have got very good at being able to put fluorescent molecules, or labels if you like, on whatever target they like. So they can make pretty much anything fluorescent. And that gives you very high specificity when you're then wanting to image your sample. You can image exactly what you want, you can do multicolour imaging and all this sort of thing. But because we're using light, this was worked out a long time ago by Abbe in the 19th century, that you very quickly reach a limit because the molecules that are doing the imaging are 100 times smaller than the wavelength of the light that's being used to form the image. So for two molecules to be well resolved, they need to be at least a couple of 100 nanometers apart, which is quite close, and why fluorescence microscopy has done quite well so far. But for the example over there, someone's gone to all the trouble of putting fluorescent labels on ATP synthase molecules, which are on the inside of mitochondria. And if all of these labels are lit up with laser light at once, they all fluoresce, and basically the whole cell just looks like a big red blob. And that's great, we can image the cell, but wouldn't it be nice if we could actually see down inside here and actually see where the actual individual ATP synthase molecules are, as lit up by those fluorescent dyes attached to them. So to go from micro world, to go from microscopy down to the nano world, we need to get around this diffraction limit. Localising single molecules lets you do this. You can circumvent the diffraction limit, and that's because we know the shape of the emission coming out of a single molecule. So on the left there, 5 single fluorescent molecules, nicely separated. That's their little diffraction limited images. And if we plot the intensity in the middle figure there, we see that they look like little witches' hats, which can be reasonably well approximated by a two-dimensional Gaussian curve. So you know the normal distribution, or the bell-shaped curve, just imagine that, spin it around in 2D, that's about the shape of that. So we can fit that, and then if the base of those point spread functions, as they're called, say is the size of that dartboard, we can locate the emitting molecule to within the bullseye area of that dartboard. So we can get its coordinates to a few nanometers by fitting that point spread function. So, imaging single molecules then, it sounds like it should be pretty hard. It's actually quite straightforward. You just need a laser, bring some light onto a sample. As long as the individual molecules in that sample are nicely spread out, and you then filter out any excitation light so that only fluorescence from the sample gets back down to your camera, which of course needs to be a really good camera, very sensitive, you can then see individual molecules. So what do they look like? If we look at these over time, you can see that the fluorescence intensity from them is not constant. So if we just have a bulk sample like that fluorescent dye in a little jar and just illuminate that with constant intensity, we'll get constant fluorescence out of that. The molecules in there are all doing different things, but there's so many of them, we just see the averaged effect. But when you look at a single molecule, you see what it's doing. Now this phenomenon of blinking, as it's called, plus localizing single molecules, lets us do super resolution. So what you do is you just have to control that blinking. Find a way to switch off the vast majority of your molecules. So you go from a diffraction limited fluorescence image to a whole lot of images of single molecules. Collect lots and lots of these images of unperturbed single molecule emissions, and then repeat that many times, and for each single molecule determine the emitter location and add a point to the final image. You will end up with hundreds of thousands, maybe millions of localized molecules to do this. So you'd think that might take a long time. It doesn't. It only takes a few minutes. And so what happens is something like this. You're doing your imaging, you're collecting molecules, you localize their position, you add a point to the image, and you build it up literally molecule by molecule, point by point. So what have we got here? We can see it coming into view now. This is a cell. You can see the membrane of the cell there at the top. That's the edge of the cell. And what we've done is put fluorescent labels on proteins called tubulin. And tubulin likes to form up into these little thread-like structures called microtubules. And so we can see all the microtubules here inside this cell. And these microtubules are about 25 nanometers wide. So that's about 1/10 of the diffraction limit. And you can see if we zoom in on these images how much better they're doing over conventional microscopy. So if we take an area there, you can see the conventional microscopy pretty much losing all the information, but there's still plenty of information there in the super resolution image. The single microtubules are really well resolved. So what are we doing with this? We've got a collaboration going with some virologists, Greg Mosley at University of Melbourne and Naoto Ito in Gifu University in Japan. And what we've seen is that rabies protein, they're interested in all kinds of viruses, but proteins from rabies virus, when introduced into cells, does crazy things to the microtubules. It actually bundles them all up into these great big cord-like structures here. And this is behind why rabies is such a lethal disease. And this has not been seen before, and we're now looking at a bunch of different viruses, including Ebola virus, to see whether they do this as well. So this is a completely new mechanism by which viruses can attack its host. Some other things we're looking at, again, mitochondria. So this is the energy source for cells. The ATP is synthesized inside, it has to get out, and the ADP has to get back in. And that all goes through the import receptor on the surface. So we've labelled the import receptors on mitochondria, and you can see that there's these little clusters forming of them, it would seem, around the membrane of the mitochondria. And that's information that we wouldn't be able to see from the diffraction limited image up there. Another little example is actin filaments. So it's again part of the cell's cytoskeleton. Actin filaments are very thin, very narrow, just a few nanometers, but they also like to bundle up into all kinds of filaments and big stress fibers like these ones here. And so the distribution of these bundles in the actin has not been seen before by fluorescence microscopy, so we can see that too. All right, just finish up by just acknowledging the students who've done all the work, particularly there, Donna Whelan, who developed a lot of this as part of her PhD, and she'll hate me for doing this, but there she is. She's sitting up the back, and she had just got the VC's commendation for thesis excellence, so well done, Donna. And of course, collaborators, and thanks to Monash for giving me some money to buy the really good cameras to be able to do this imaging. Thanks.